I'm Peter Kine, a research fellow at Charles Darwin University. I'm leading a project for the marine biodiversity and Northern Australia hubs of NERP. We're focusing on a variety of threatened euryhaline and elasmobranchs. Elasmobranchs are sharks and their relatives, such as sawfishes, stingrays, manta rays, those sorts of things. And euryhaline refers to species that can live in both fresh and salt water and move freely between the two. Here in the South Alligator River, we've got one species of sawfish as well as 3 species of sharks. The northern river shark and the spear-tooth shark, both of which are threatened and which we're working on, as well as the more common bull shark. They're listed under the Commonwealth legislation, the Environment Protection Biodiversity Conservation Act. Being species that are listed on the Act, they require recovery plans to be implemented to guide and direct their recovery, conservation and management. To be able to do that, you effectively need to know what the population status and size is so you can measure conservation activities against that. What we're trying to do here is map the population by looking only at the juveniles and subadults that we find in the rivers. Today we're continuing on with some monthly sampling we're doing in the South Alligator River. We're investigating the populations of these 2 sharks and the largetooth sawfish here over the course of the next 2 years. We hope to look at how they're utilising this river system, the movement patterns, the population structure, what size classes, if there's these young sharks that are using the system up to subadults, if we can detect the movement outside of the estuaries when they move to sea. And we're putting all that information together to look at the overall status of the species within these systems. We're using a gill net to try and capture both sharks and sawfish. This is a net that's very effective at catching these species. So we set that net and we look for movement of our floats to indicate that we've caught something. We regularly check the net. If we have caught a shark or a sawfish, we remove it from the net onto the boat, and there we can start to take a series of measurements on the individual shark or sawfish. We take a genetic sample, that's a small tissue sample, just fingernail size, and that's used to extract the DNA, and that can tell us a lot of information about that fish and about the species as a whole. Each fish is tagged with a tag individual number, so if we recapture an animal, we can see that we've had that animal before, and we can also look then at its growth rates and its movement and its residency times and those sort of things in the river. Both the spear-tooth and the northern river sharks have restricted ranges in northern Australia, and about half of their known locations where these species are recorded are in Kakadu National Park, in pristine river systems such as the West, East and South Alligators. That protection within the park is probably what's allowed these viable populations to persist here. Through our research, we're showing that they're quite important nursery areas. That's where the young sharks are spending the first few years of their lives before they head out to sea. When they then move out offshore, they face the risks and threats that aren't present in the park in these systems. So we need to look at what interactions may be occurring outside of Kakadu National Park when these species get offshore with commercial fishing, those sort of things, and where they're moving to. So we don't have a good understanding if there's exchange between locations that may put them at risk from being caught as bycatch in other fisheries that occur around the region or more widely. So we hope that this monthly program of sampling will help to determine whether existing management strategies such as retention bans are effective, and more importantly, what sort of other management might be required to ensure the survival of these species.